Good afternoon. Today I'm going to be covering the case of the clever neuropsychologist from the Biopsychology text in Chapter 11. The case focuses on episodic memory deficits as well as the ability to induce false memories. I'm going to be covering the neurochemistry and the brain structures involved in memory creation, maintenance, and retrieval. And then I'm going to be looking at the ethical implications involved with inducing false memories. So I chose this case study because to me it challenges how we historically have thought about memory. Many of us tend to assume that memory is very accurate and is a very reliable facsimile of events that have happened in the past. However, this case shows to me that memory is in fact very fallible and it can be manipulated to different in different ways. I also found it very interesting how false memories can potentially create different clinical therapeutic methodologies. And through manipulating memory, we can change the way that we actually treat patients. Um, for me, the idea of false memories and memory manipulation has serious implications for biopsychology and for medicine, as well as even the legal system. So I'm going to be taking a look at that today. So, case study overview. The patient had difficulty forming or recalling different episodic memories. Despite this fact, the neuropsychologist was able to influence what the patient remembered. Um, the clinician did this through different questioning and through suggestion methodologies, such as leading questions. And the patient began to recall events that actually never occurred or never actually happened to them. This demonstrates how memory is reconstructive rather than a perfect recording. So the memory is rebuilt every time it's retrieved, rather than it being a existing facsimile that we go and we grab and we look at as a perfect copy every time. So here in the image that we're seeing, it's depicting a brain fMRI through different memory tests. And what this image goes to show is the various brain structures and their disparate locations and how they're all working simultaneously to retrieve different parts of information and then pull that together upon retrieval. And so the patient presented with episodic memory deficits. Patient could not form or retrieve accurate personal life experiences. The neuropsychologist used suggestion and questioning to influence the patient. The patient began to recall or form false memories. Okay, so what is episodic memory? Episodic memory is a type of declarative memory. It's memory that we actively declare to ourselves that we're going to go and retrieve and withdraw and pull that information. It typically involves personal experiences that include things like time, place, emotional context, and it's different from semantic memory, which usually involves like generalized knowledge. And so episodic memory is very vulnerable to distortions and alterations, as we'll see. Episodic memory, in order for that to be formed, it has to become a long-term memory. So we have a stimulus. That stimulus activates our sensory memory, and through the repeated activation of that sensory memory, it can then become a short-term memory, and it can then, from short-term memory through long-term potentiation, be stored as a long-term memory, which we can then actively retrieve. And so this is really interesting to me, especially being a student, wanting to learn about the process of episodic memory and also, semantic memory to store information and then actively retrieve it, for the purpose of classwork or for the purpose of going on through my academic career. Um, So the image that we've got here is one of the different brain structures. All of the brain structures here, identified here aren't necessarily included in the activation of memory networks. However, this does give us kind of a generalized roadmap of the brain to work with to identify where these structures are at in relation to other structures. So the prefrontal cortex is kind of the portion of the frontal cortex is the portion of the brain that's at the front or anterior of our anatomy. We then have the parietal cortex, which is kind of a sensory motor area. It's kind of like it's kind of dorsal and posterior, if you will. We have our temporal cortices, which are lateral to our brain anatomy. And then we have our occipital cortex or occipital lobe, which is posterior and kind of ventral, if you will. And so the frontal cortex or the frontal lobe is responsible for higher cognitive functions. It's kind of what makes us human and our ability to make decisions about our life, to think about the future, actively recall the past, and it's very, very pivotal for retrieving memory. Our parietal cortex is kind of like sensory motor, but it's where the sensory information comes in, and it has a lot to do with kind of like muscle memory, if you will, as well as just transducing sensory information to then store it in other brain structures and locations. Our temporal cortices, or our temporal lobes, are very, very important for language, speech production, and speech interpretation. And also, as a function of that, they're very important for memory. As you might assume. And then our occipital lobe is responsible for processing visual information, which is also very important. When we see something transpire in our lives, and it has emotional weight to it, then we may store that visual sensory information as a memory in our brain. And so all these different structures are responsible in some part for helping in the production, maintenance, and retrieval of memories. The structures that are heavily implicated are the hippocampus, which is responsible for some of the memory consolidation. It's critical for forming new memories. Our amygdala adds the emotional weight and the emotional significance to our memories. The prefrontal cortex, like I said, it helps to organize and retrieve the memories. And the entorhinal cortex acts as a relay between the hippocampus and the cortex. And so damage or dysfunction to any of these areas can lead to severe memory deficits. So something that's really, really important in forming memories, retrieving memories, and the long-term potentiation or turning a memory into a long-term memory is the different neurochemicals involved, or the neurotransmitters. And so there are a few that are heavily implicated. Acetylcholine is a major player in memory encoding, and it helps us to actually give conscious attention to specific memories, so it helps with that memory retrieval. Glutamate is an excitatory neurotransmitter, so it helps us to activate the synapses and the synaptic pathways to go in and actually pull memories forward and retrieve them and give the conscious attention to them. GABA is actually inhibitory, so it kind of counteracts glutamate, and it helps to maintain balance in our neurochemistry, so we don't overactivate a neural pathway. Gamma aminobutyric acid is a type of amino acid as well, and so we'll talk a little bit more about amino acids here later on in the presentation. So just keep that in mind. And then dopamine helps to reinforce memory, and reinforce specifically memory salience. It's really important for the emotional significance and the emotional impact of memories. So to create a memory, we have to take a memory or a stimulus and put it through the process of long-term potentiation. And so what this means is that the efficiency and the ability for a synapse to activate an action potential on a postsynaptic neuron becomes better, or it strengthens over time through different mechanisms. So those mechanisms might be increased neurotransmitter release, or an increase in neurotransmitter binding sites on the postsynaptic neuron. And so this can happen through different receptors. For example, we have NMDA receptors, which help to enable long-term potentiation. NMDA receptors are essential for synaptic plasticity. They allow calcium to enter neurons, which triggers learning processes. You also have AMPA receptors, which strengthen synaptic transmission. And together, these two receptors can lead to long-term potentiation and help to support that potentiation over a lifetime. Embedded in the presentation here is a video on synaptogenesis. I wanted to include this video because it helps to go to show how the brain forms new neural connections with dendrites and axons. And this is necessary for forming new and robust neural circuitry in order to help to potentiate long-term memories. I can't play the video while I'm recording, so I'll just leave it embedded here within the presentation. So again, I've got another video embedded here, which describes enzymes and how enzymes catalyze a chemical reaction, meaning that they help to speed up a chemical reaction. They help to lower the energy cost to initiate the chemical reaction without actually being consumed in the reaction itself. And a lot of the processes that go on in our brain wouldn't actually be able to happen without these enzymes to catalyze those reactions. And so enzymes are proteins or polypeptides that are made up of amino acids. GABA or gamma aminobutyric acid is an amino acid, and so that's why I wanted to kind of come back circle with that. But these amino acids are proteins, and or they're peptides. The enzymes are proteins or peptides made of amino acids, and this is important because through different gene expression and epigenetic effects, we can have an increase or a decrease in specific enzyme activity, and so this can help or hinder memory formation, maintenance, and retrieval efforts. And so, for example, we can get protein, we can get histone acetylation or methylation, in which different segments of DNA become more or less available through those epigenetic effects. And in them becoming more or less available, we may have more protein kinases being formed. And so, for example, protein kinase helps to phosphorylate or transfer a phosphate group from one molecule to another. And this is very, very important for energy expenditure. ATP, our primary energy currency, uses phosphate, and so by adding and removing phosphates to ATP back and forth to form ADP is how we actually, use energy and make things happen in the brain. And so if it wasn't for these different protein kinases or these different enzymes, we wouldn't actually be able to do any of the memory forming or retrieving efforts that we do in the first place. And so it's very, very important that we recognize that enzymes have a important process in DNA in memory formation and potentiation. So within the case study, there was an emphasis on the ability of the clinician to create false memories in the patient. And so memory is reconstructive. And this is shown by the events of that false memory. When the brain is trying to retrieve a memory, a declarative episodic memory, it doesn't take a perfect facsimile picture, snapshot, if you will, of the event that you're trying to retrieve and bring it back to your conscious perception. What it does is it takes different encoded information associated with that time and place and people and objects, etc. It takes that associative encoded information and reconstructs it, puts it back together, and says, this is probably a good amalgamation of what actually occurred. And as a result of this, you can alter it every time you recall it through suggestions and leading questions. The brain will try and fill in gaps in the memory with plausible information, even if that information is incorrect. And so this can lead to different errors, such as source monitoring errors, where we don't actually know where a memory came from, and we may even perceive the memories of others as our own memories. So there are several different experiments that were done that help to support the case study and the theories that it's posing. One such example is research done by Elizabeth Loftus. She demonstrated that it's pretty easy to implant false memories. In one study, participants of hers were led to believe that they were lost in a mall as children, and many participants later recalled the event as if it were real, even if it wasn't, which this goes to show how powerful that suggestion can be in shaping our memories. It really, really shows that memory is not, it's not like cement that's solidified over time. It's more like steel or plastic, where it can actually be changed and reshaped and reforged into something new every time. So there's a lot of ethical implications around false memories and even just the plasticity of memories in general. Patients could develop false beliefs about their own past, which can lead to emotional distress, false accusations. It also creates a risk of misuse in therapy or interrogation. Clinicians could inadvertently or purposefully lead patients to a false belief. And so clinicians should avoid leading or suggestive questioning, and they should use evidence-based interviewing techniques. Um, Patients should also be informed about the limits of memory reliability, as well as specific um, clinical therapies. They should be well informed of the possibility that they could be accidentally or inadvertently led to a false belief. Also, it's really important that we have ethical oversight to prevent possible and intended or unintended misuse. I learned a lot through this specific case study. It was really insightful for me. It showed to me that memory is really not a perfect recording of historical events, but it's an amalgamation of approximations, if you will, and it's heavily influenced by brain chemistry and neural activity. It's shown to me that memories can be altered by external factors through like suggestions or leading questions, or even just having ambiguities in our memory can allow the brain to fill in those ambiguities with incorrect information that just might make sense, but is not actually truthful. Um, and so I'm a lot more cautious now about trusting recalled experiences, and trusting things like eyewitness testimony or even the personal memory of others. And it really goes to show that there's a heavy importance of scientific understanding in how memories are formed, maintained, and retrieved, because there are a lot of different implications, whether it's medical or legal or emotional or what have you. And so yeah, this was really, really interesting for me. Key takeaways for me is, memory is malleable, it's reconstructive, and it's oftentimes very unreliable. And there are ethical associations with that, in that patients could be misinformed, patients could be led to a false belief. There are legal implications when it comes to testimony in court, and there are medical clinical research implications as well, and that it creates a situation where it's now harder to separate fact from fiction when it comes to our patients and what they may have gone through in the past, whether it's treating some disorder that happened at some point in their past, or whether it's helping them through through therapy, overcome something in their past. There's different nuanced challenges that arise through lapses in memory or false memory or memory deficits. And especially being a student and wanting to find the best way to learn and remember my coursework, this was really, really interesting for me to cover. And so thank you for sitting through the presentation. I hope you have a wonderful day.